presentation about horizontal gene transfer for transfer. Well, it can be transferred by viruses, but for transfer genetic material, of course, I think we should have a social communication, a way to interact. And in the case of capitalism, I was wondering if this rare biosphere, they are alone, they are in microcosms, which are the consequences.
The students, does anyone have questions that arise during the talks of the speakers? Things that they don't understand, that you want to clarify? It's our new student.
Maybe that I could understand. But one thing that I find intriguing is that these very abundant bacteria like SARS-11 and protocols and silicotrophs, their virus is known for them. And I wonder how they will survive in the oceans being always abundant. So it would be nice to know whether some experiments have been done with continuous cultures or like they did on the research of
Well, I will use a microphone. We can't hear where.
You don't,
You know,
¿Cuál es el micrófono?
other questions, comments? Yes?
There was a few speakers who were in a project in biodiversity, biotechnology and bioinformatics, I think. So how they think the knowledge about biodiversity could make an impact in the several problems we have today about impacting, climate changes and acidification and several problems we have today in the oceans.
And do you think this is a question for all the speakers or some specific one?
I mean, yes.
Okay. I think we have several topics. Some of them are very general. I will also try, or wish to include, I will also wish to include the further steps that we can imagine in the career or in the dreams of Carl Busick. He will alive today. What will he dream to study or to advance in? That would be my question for I think for other speakers. So I think we will start with the more focused question one. That was, for example, I will try to summarize all this. So, yeah, let's start with the question from Carlos to Mark about the virus from the SARS-11 and the advances that we have about that. I think that we should.
What are the advances?
No, the question was whether experiments in continuous cultures, such as those that were done with E. coli and B viruses, have been tried in cyanobacteria.
The cyanus, so not Okay, so well, I think that is two questions. So there are experiments in Jennifer Hughes Martini's lab on cyanobacteria and viruses, and they've done some nice work to show that resistance can evolve in communities, and they've narrowed that down to particular genes. And strikingly, it was not genes we would have predicted a priori. We thought of BTL fibers as the easiest first thing to mutate. They would change the virus to alter host range or surface proteins, but it ended up being much more complicated than that and involve So that's the example of cyanobacteria being used in chemostats to look at virus host pressures. It's not been done to the depth that was done in the 70s with E. coli, but that's because of the challenges of cyanobacteria. None of that's been done for SAR-11. And the work in SAR-11 is, there are 4 viruses that are published, and Stephen and I are actually working together on applying viral tagging to Lagibacter to try to just map in a couple of key sites what virus sequence space looks like and get a handle on the total viral population for Lagibacter before deciding too much on what we might want to focus on. But that, I think that's about all I can say about SAR-11 viruses. What changes in the virus lead to changes in host strategy? So she was sequencing the viral genomes.
So we don't know whether the cyanobacteria become resistant.
We know that cyanobacteria, any culture you leave on the shelf for a while will show spontaneous resistance. It's very little investigated at a mechanistic level. In fact, I can't think of any example actually characterized on a molecular mechanistic level. But it happens every time. Just leave the culture. It'll be green. Or orange or whatever color you're setting on this.
And you can sequence single virus?
So there's one paper in 2011, in class 1, that sequenced a single virus. It was in Lambda as a test, and nobody's revived that technology. The particular lab switched careers to go save a really beautiful lake in Idaho, actually. And I'm not sure when we need to do the single viral genomics work for this level of stuff, but there is a technique out there that's a little tested and has been done once.
I know that you mentioned during your presentation about the RNA virus.
Can you speak louder, please?
Yes. I know that you talked a little bit in your class during the language about the RNAs, RNA virus, and during your talk, if maybe I didn't mention, but I think you didn't mention them. So I would like you to elaborate a little bit more about this virus and why they have not been looked with such emphasis like the DNA virus.
So the question is I hinted in the class about RNA viruses maybe being a bit important player in the oceans, and I didn't today. Why? So there are data recently from Greg Stewart's lab showing that if you concentrate viral particles and extract nucleic acids, make an assumption based on the nucleic acid you've extracted and genome sizes, then you'd suggest that about half of viral particles are probably RNA viral particles in seawater. I'll tell you those numbers range from 17 to 72%, and they looked at 2 sites. So that's all we know. RNA viruses are probably important. They're probably important in eukaryotic phytoplankton. Why does no one look at them? Well, Alex Colley's looked at them using targeting the RDRP gene. It's highly conserved enough. RNA viruses mutate very rapidly, so having your partner sets as they are. They're small, so I don't look at them because they're boring. Very small genomes are just, they're not particularly exciting for me. But I'm sure they're really important for people who would study eukaryotes. Yeah, they're probably really important in evolution. I also don't look at them because my hands are plenty full of just the DNA viruses.
There was another question from Kurt about mass viruses, genes transfer in the ocean. What do you can comment about that?
So if I recall the question, it was, what is the mass of a virus? And my answer would be that's a lookup. I don't memorize the lookup numbers, but what actually makes?
Yeah.
When the whale is being broken down by bacteria being broken down by viruses. I don't know what eats viruses at the end or their carbon but it's also loss of energy very important.
That's right. Yeah. So those numbers have been calculated, and it's like 50 million blue whales. You know, there is a calculation out there. It's a big number. It's a lot of blue whales. Maya Breitbart and Forrest Rower, I know, put a number out there. Do any of the rest of you remember the virus whale conversion? and therefore they're not important in horizontal gene transfer. That was the...
Not therefore. but,
Okay.
Because of the unlikelihood that real metabolic pathways would ever be completely transferred.
Yeah. Well, I think there's two flavors. So the question is really, I was suggesting that it seems unlikely to me that whole metabolic pathways could be transferred. And I still think that's true. Norm made a good point that there are examples of metabolic pathways where the genes are clustered and they're 40 kb-ish, so we could imagine them being transferred. And I think that those could certainly be the case. But evolution isn't just whole metabolic pathways. It's also turning the knob and tuning gene sequences of individual genes in a pathway. And, Paul Fikowski said, small changes in a tiny little gene can affect planetary chemistry. And he meant that at the level of the viral PSBA gene. He meant that at the Rubisco was his big case. So I think you can imagine viruses could move one gene for their own interests accidentally, just by the way this sloppy evolutionary process might work, and that could impact a metabolic pathway. Now, of course, it should be, in most cases, detrimental, so selected against. But at 10 to the 29th transduction events per day, some small fraction of the time, there might be a net gain and a fitness advantage in a fixed event.
Can I make a point?
Yes.
Just a point in passing. We talk all the time about the transfer of genes, transfer of functions. Let's remember that it's not only transfer of genes, transfer of functions, it's transfer of sequences. And we know many examples where patchworks of sequences have been transferred, have been used to construct other things. And a good example of that would be two-component systems. You can see two-component systems in different organisms which have different regulatory components. And it's clear that one component is related to the other, but only over part of its length. And the other stuff came from who knows where. So I just want to make the point that it's not only, don't think it's only transfer of functional genes, but also transfer blocks of sequence, which can be compiled into Legos.
Just stay here, Norman, because we have also... Yes, we can try to... No. Can you comment something about the innovation in technology and the arising of this NGS and the coming out from this length, ranked width from short to length?
Yeah, well, the point of long sequences is having accurate phylogenies. The accuracy of any particular phylogeny. I'm interested in understanding the structure of the tree of life. And we can do this with ribosomal RNAs at some level, but you're still limited, pretty limited, to what you can, the number of characters that you have. And particularly, as I mentioned, the deeper you go into a tree, the more uncertain the branch points become. One approach to alleviating that problem a little bit is long sequences. Now, there are two possible things. One, you can sequence all of the small subunit or large subunit ribosomal RNA genes, but it still may or may not get you there. Another possibility is to use concatenated genes, and there are many people who are stringing together concatenated genes and trying to use those to infer phylogeny. I don't like that process myself for several practical reasons. I have no theoretical problem with it. The practical reasons are what people do when they download, when they construct a concatenated gene group to do phylogeny with. They download sequences. For example, the ribosomal proteins are probably the best for that. And so people will download ribosomal proteins. They'll do some kind of automated computer alignment. Bad start, bad start. And then they'll use these things with no attention to differential rates of evolution. And so you pile uncertainty on uncertainty on uncertainty, and I'm not convinced you're getting a better answer from a concatenated ribosomal protein story. In fact, I think you're probably getting a worse answer than if you use full-length small subunit ribosomal RNAs. Now that at some level is a philosophical statement. The problem is this issue of homology. So critical to a phylogenetic analysis is lining up the sequences, counting the number of differences. But the important thing is that it's important to know that every sequence that you include in that calculation is a homologue of common ancestry. And so when people download sequences and do the computer-based alignment, I know that different alignments give you different alignment programs give you different answers at some level. But that's a level which introduces confusion. It introduces not good information. And then you fold in things that we don't understand, like rates of change of the different genes. We know that the different ribosomal proteins change at different rates. We know that regions within the ribosomal proteins change at different rates. So you don't have any way of handling what's changed.
Okay.
So my problem is not necessarily theoretical, but it's practical, the way that people do it.
Okay. Okay. Thank you, Nor. Sure. So we have also some more broad questions. No, we still have a question for Linda. That is a specific, I think, question about the biodiversity impact over questions from changing oceans. And I think we can also mix with the use of classical ecological indices that Carlos also asked.
Carlos? Oh, Carlos. Oh, sorry. So when you say classical diversity indices, what does that mean to you?
To me, for big animals or mix of diversity and
Evenness and
Richness,
yeah.
alpha diversity, beta diversity.
Well, you know, it's a point well taken. The indices that we've been using, that I've, I guess, been using, but also ICOM, are actually newer, newly developed. I didn't stress this, but the parametric methods are model-based methods, and they provide confidence bounds for an estimate. It's argued that when you make multiple comparisons, that your confidence bounds get huge, which is true. But it's actually a difficult problem, and I think that a bigger part of the problem is that statisticians aren't interested in it, quite frankly. So the people that are capable of really facilitating the field are not so interested in the problem because they don't find it intellectually challenging. And this is what my statistician friends tell me. So that's part of the problem, is we probably do need different ways of thinking about this kind of data, but the sort of the thinking and the people that are doing the thinking aren't on board yet. Hopefully that'll change in the future, because I think that, you know, one of the things that these large-scale projects are doing are perhaps in making people that have made a career out of ecological statistics and are interested in these kinds of questions more interested in being able to help us answer these questions and get reasonable estimates. So that's my answer.
Okay. And how can we link this with this biodiversity analysis in an overall context, and thinking in a changing, or all the problems that are facing today?
Is that the second question?
Yeah.
Yeah, okay.
Sorry, I will try.
Yeah. So I thought, so there's some interesting things I think that are developing with respect to richness and hotspots. Classically, we've sort of always considered the tropics as the hotspots for biodiversity, right? But there's been some paradigm shifting, I think, if we can believe the results, that perhaps those aren't hotspots for microbial diversity. But then again, if everything is everywhere really, you know, does it matter where those hotspots are? So I'm going to answer my third question, which was something about what are your big questions? Was that Oswaldo's?
Yeah.
What are the big questions you want to ask?
Yeah.
Okay, so here's my big question that I really want to ask, and that is, are there regime shifts in microbial communities that will have irreversible effects? We keep talking about microbes as infinitely resistant or resilient. Is there a tipping point in microbial communities such that, you know, the environment might change and those critical microbes might not come back? And you know, what does that look like, and when will that happen? That's my question. And hopefully I've answered your question, maybe, about how you can use this information. I mean, we can draw these maps, great, but, you know, again, we don't know at what point, when things become so richness poor, I don't know what the right word is, but deplete of species, you know, at what point are there still enough members in there to do all those critical functions? You know, I like the example of expanding HABs, harmful algal blooms, as an example of okay, we know that there can be changes and shifts in communities that do have an impact, and it's a real impact on human health, for example. And that, I like that example because it speaks to the fact that, okay, organisms are still there, they're still present, but their numbers, their relative abundances are increasing, and that's going to make a difference ultimately. And that's, I think, a good example of it's not just being there, but if you're there in high abundance and the system can't cope with it, then it could ultimately have actual impacts, measurable impacts on industries, such as the shellfishing industry, et cetera. So those are the kinds of impacts that maybe we need to pay attention to, but there are going to be other ones that perhaps are not so obvious that we won't know until it's too late.
Okay, thank you. I think we can try to continue with these big questions that will arise from here. Maybe Carlos will tell us. Yeah, you. Please. What do you think are the big questions that are not yet, or that need to be addressed in the future?
I don't know.
Okay, the next speaker, please.
there is one question that I would like to know the answer for, and that's that with the genomics and with lateral gene transfer, I think we're realizing that our ideas about species and evolution are biased by organisms of our size. So we think species are these discrete units, and that evolution is functioning because these discrete units interact and they compete and this kind of thing. But I think seeing how all these pan-genomes have so many different genes for what we consider to be a species, and seeing all this lateral gene transfer, how easy it is to exchange genes, I wonder what kind of idea of what a species is will we have in 15 years or in 20 years. But I think it's going to be very different. And I think there are going to be mechanisms there of evolution that we haven't seen before, because probably they function only in microorganisms and they don't function with animals and plants.
Can you comment on the question?
Of course.
But so there's, I actually want to talk.
This is the...
It's not controversial,
Please.
I just would... So there's a recent review, Bill Hennick and Martin Poltz and Eric Holm, that I thought captured a lot of a very complex, confusing topic and put some things to clarity for me. So they basically posit, and this was vaguely mentioned earlier, that some genes are transferred often and other genes are less transferred. And when we talk about a pangenome, there's probably certain kinds of genes which are more often transferred and flexible. And I used to think of RNA as never, and tRNA genes perhaps as often transferred, right? And yet there's probably this huge gradient. And so out of that, though, still emerges clear species definitions that fit classical species concepts that are okay from the real world. And I wondered if you could comment on that paper. Did you see it? It was in Trends in Genetics in last year. Okay. So it's about 6 months old, so I wasn't sure. Read it, and then send me an email.
I have also a question about that, because arising from this pangenome theory, there is also a more speculative theory that talks about the hologenome theory of evolution, that try to say that in fact that what is evolutioning is not a single genome, that is in fact the combination of several genomes that are interacting, but in fact there is no single organism that is living a part of the other. What do you think about that?
Well, that's, you mean, that's something, I think it's related to this question of the pangenome, right?
Yeah.
So.
In the lateral transfer, and then in fact there are all together the.
Yeah, I think I need to read this paper before continuing with this topic. But I think it's not easy to define species even for birds, which they are so easy to study and everything, and yet it's not easy. There are at least more than 25 different definitions of species for large animals and plants, and I don't think there's any good definition for bacteria. There's these pragmatic things that we use all the time, but all this flexibility and all this interaction among different genomes, I think it's going to be. At least I have difficulties thinking of how this functions in evolution. So I have to read this paper. Maybe it's the solution, and it's not a question for the next 15 years, but it was already answered. I don't know. But the really important question for me is not a microbial ecology question. The really important question is to understand why in the 19th century slavery was abolished. This is something that I'll leave you to think about, but it's something that really fascinates me.
Plop. So thinking or going into more detail about the next questions, Norm, can you say something about that? What will?
Species?
No, no. About the question that must be addressed, in which we have a big gap, or do you think that we could advance in?
I think there are many things that I would like to know and like to see advancement in. I think if you ask a very fundamental question, my question would be a little bit vague, but on the other hand, somewhat precise. What are the mechanisms of evolution? Now, this is not a trivial question, because we traditionally think of evolution of being Darwinian evolution. Our models are all based on Darwinian notions of evolution, almost certainly incorrect. Let me give you an example of what I mean by this. So Darwin posited that evolution was a slow course of slow change in which one species of organisms, which we have to return to that definition, where one species of organism turns into another organism. There is no example in history, in geology, of one organism turning into another organism. This has been known for a very long time. It was pointed out by Stephen Jay Gould and Niles Eldredge in 1967, the concept of punctuated equilibrium. The idea is that if you, for example, take sediment cores, deep sediment cores, and now start looking for clams, for example. You start at the bottom and no clams, no clams, no clams, and all of a sudden there's a bunch of these damn clams, a bunch of them, rudimentary clams, which are indeed honed on with further change, but that change is a variation, not speciation. There is no example in the geological record of one organism turning into another organism, and this has been shown. And the other thing is it even possible for one organism to turn into another species? I don't think so. Now, any particular species, whatever that means, any particular species can be a very complex collection of organisms. The definitions of species traditionally have been based on interreproducibility of the same members of the same species. Now that works with animals at some level. It falls apart in plants. Hybridization, right? And that's an example of a non-species to species transition. Where was I going with this? Carl Woese had an interesting analogy for species. And this is, if you're looking at a stream flowing past, the stream will have eddies, and the eddies will persist. Even though they're not physical, they'll persist, and then maybe go away. That maybe is a better analogy for evolution than species turning into species. So what is that mechanism of punctuation? Can I draw a picture?
Yeah, sure.
This all boils down. This all boils down to. Good. So there are two modes. There are two modes of evolution that have been proposed. One of these modes is the traditional Darwinian approach of the molecular clock. And this would change, so if we had accumulation change, and this is time. So the molecular clock concept, Darwinism basically, would be the rate of change is constant with time. And there are people who will try to use the number of sequence differences to get some idea of time. Now that works maybe over the last 500 million years, but it doesn't work if you go deeply. Then the alternative way to the Darwinian approach is what Gould and Eldredge call punctuated equilibrium. And the notion here is that when organisms are, they will slowly change by variation. But variation is not speciation. Speciation is something special. And then something happens and there's a punctuation, so there's a lot of change accumulated very fast. And then it settles down again. The sequences are always changing, always undergoing mutation. There's no question that this change happens. We'll call it equilibrium, but it's always changing. The important thing is that the punctuations happen again and again and again. And very early on in evolution, if we go back now to the beginning of zero of life, the rates of change have probably tailed off a lot, coming out to where we are, certainly. So, I would argue that what we observe is punctuation, not constant clock, not any sort of constant clock. What are the mechanisms? What are the mechanisms of these punctuations? What does that mean? How does a new collection of genes come together to give a new organism, not a variation? Darwin's finches were not speciation, that was variation. And in fact, a lot of animal and plant speciation is variation, not speciation, probably. One would argue. So I think these modes are very different, and the mechanism of that punctuation is critical to understanding evolution. So there's woes as eddies that brings those together and creates it. I don't think we know.
Would you say no species evolved from other species also for bacteria?
I think that's correct. And the problem with bacteria, of course, is that they're so manifold in the sorts of properties that they have that you might say, oh, well, this rhodobacter that doesn't have photosynthesis, because it just doesn't have the genes yet, that's rhodobacter. And another rhodobacter is photosynthetic, it has the genes. And there are a lot of organisms, phototrophs, non-phototrophs, they're very, very closely related, which exchange genes very freely, probably, for example. But it's not speciation. Not to say that I know what speciation is, but the idea of the species being defined by reproduction, that really falls apart in places except animals. But in terms of thinking of the reality of the evolutionary process, it is not incremental, it's punctuated. And the remarkable thing is, if you look at the book in evolution, any discussion of punctuated equilibrium at this time, the books that I've looked at, tend to be a side box, not the truth. Side box, not a fundamental thing, but a side box. Isn't that interesting? Because we don't understand it, of course.
Okay. No, it's okay. So the big question would be about the mechanism of evolution. And what do you think, Daniel, for example, about
the mechanism of bringing together a collection of genes that can function?
What do you want?
I was thinking, sorry.
I'm not. My background is more engineer background. Actually, I'm not a biologist. So for me, some of the question, and actually you quoted in your talk, I really like to say that in order to understand things, we need to put order. Order is a key, actually. And sometimes when I look at some of the data we're acquiring, for example, with all these metagenomes or all these tag sequence, from the all over the world, I feel that we're still missing a lot of order. Things don't make sense. People are doing, these clone libraries, or every study, so before it was clone libraries, now it's tag sequencing, they find this alpha proteobacteria, this beta proteobacteria, but to me it doesn't make real sense. I mean, where do we find, a group, like there are some groups where picture begin to emerge. So my feeling is that I will not go for the big picture, try to build some kind of image of the biodiversity at the very large scale, but my approach is to go to each of these groups and then tweak things apart, because some of the groups will begin to understand more or less their metabolism. I think there are a few good examples of where we begin to do this. One very, very good is Prochlorococcus, I think. But when you look at the number of people who work with Prochlorococcus, not that many, but I think they have done a great job actually at defining some kind of niche for each of the ecotypes, whatever you can call them. They're not species, they're ecotypes. And then try to begin to see where you find them actually in the ocean. And for me, it begins to make sense. I think for some of the eukaryotes, we begin to have that. I mean, we had that for diatoms, but still, I think it's, or dinoflagellate, but I think it's still very, very fuzzy. But for some of the, what we need is to target things that are widespread enough. Because if we look at a very specific species which is in a single habitat, it doesn't make much sense, or we just do very ecology, like for example for this, I think in this extreme environment also we can make sense of things. So I think we need to raise more models that we study in very much details. I think, for example, the mass 4 is another good example, because something that we find almost everywhere. We can begin to understand, I mean, why we find it there and not there. What are their prey, the predators? So I think we need to reconstruct the breaks and make sense. And also, there is one dimension that we didn't mention here because we still don't have the tools to link biology, but is the physics, of course. And I think this is cruelly missing also to understand in environments how the distribution of the organisms that we find match with physics. And I think we can do this if we focus on very, very specific groups of organisms. Then we can begin to make sense. So for me, at least in my approach, I think some of the progress also it's not big questions, you know, it's not everything is everywhere, or it's more, I think a lot of the progress will be made through this. One question I have actually for the other speaker is, so we had, I think there has been a couple of paradigm shift. I think in the 80s there was the discovery, for example, of the very small cells. Before we had this linear view of the marine ecosystems, from diatoms to the whale. Then we began with the microbial food web, we began to have a new concept actually. Then began the approach that Carl and Norm developed with looking at the non-culturable part. So now we have this metagenome sequencing. Okay, so what's next? That's, do you have any idea? Because I don't have any clear. And what you have to see is that most of the time, this change in paradigm came with new techniques. But like really the change, for example, discovery of phytoplankton was epifluorescence microscopy, a technique which was used elsewhere but not used at all in marine biology or marine oceanography. Same for sequencing. I mean, sequencing had been done before. I remember very, very well. I was still, I just finished my thesis with Penny, and she was taking the Woodsall course in microbiology, or and she was explaining me that it was great. You could do primer, PCR, and say, what are we going to do with this? And now everybody is doing it, of course. So are there things out there in the field that we don't have any idea for and that are going to change our view of the ocean? Do you have any hint for that? I mean,
can I say something to that? Maybe that can be related with the single-cell genomics technology that is arising now.
But
maybe
this is just a combination of you know, flow cytometry and genomics. There's nothing new in single-cell genomics. I think the idea. I pay tribute to my advisor, Penny Chisholm. But I think when I was doing my thesis, she was telling me, you know, always think from the point of view of single cell, what a single cell is doing. And single cell genomics is just that. I mean, so there is nothing really new there, just putting technology together. But
it's not a new technology.
No. What I'm saying is something maybe coming from astronomy, coming from nanotechnology, from you know, that could actually I think the NGS sequencing came from some connection with nanotechnology in some way. I mean, that's so. Is there? So I have the question for the
audience.
Yeah.
Matt,
you have the answer? No.
Well, I don't have the answer, but certainly, so if I do a simple calculation of the number of viruses that I think infect a certain host, and how long I think those viruses have to survive, they should never find their host. That tells me that the sea is not homogeneous, and there are probably microscale patterns. There's patchiness in the sea. So I see that as microfluidics being a great tool that is going to help us learn a lot in the next 10 years. Farouk definitely, Roman Stocker, yeah. I mean, microfluidics is there's some, you know, I saw Roman give this great talk of, he was following coral flagella, and he says, look, there's this microbe which spends so much energy bursting all of a sudden, and it's a pathogen, bursting all of a sudden to make this move, tons and tons of energy. But it can't move very far. You know, why is it going to spend all this energy all of a sudden? And then he did a microfluidic study with imaging to show that it is a complete perfect storm right next to the coral, because the coral has these flagella beating, and it's creating this massive, incredible vortex. And so this pathogen has this time, and it's all of a sudden it's got like probably microseconds to get on that coral, or it's gone and lost, right? And so it spends all this energy in this incredible sprint because of this. And that's fascinating. And there's going to be so many examples of that, I think. And microfluidics coupled with the powerful optics, it's just coupling two new technologies. But it could really help change the way we think about the way a microbe sees its environment.
Thank you. Do you want to say something?
Well, just yeah. I think one area I think where is probably going to be a lot of progress, but I see that things are beginning to go this way is all these interactions. I mean, which we have to find new ways actually of. Like, I mean, this, for example, you were showing during your lectures these beautiful pictures of Ostrococcus viruses interacting with the host, and for this, I think we have to. I remember, Carlos, you gave a talk in the last Gordon conference about this avalanche of sequence, if I remember well. And I think that's one of the things that we have to maybe also get away from this avalanche of sequence at some point, because it's going to really submerge us. I think it's already submerged me because I don't know where to go. I see all the sequence. Wow, wow, wow. Wow. And I think we have to really get back to also old techniques. Like what you are developing, for example, or in our case, for example, we are doing cultures, and for a long time we use like sophisticated, flow cytometry sorting and so forth. And now 90% of our culture are single, this pipette, glass pipette. Exactly. That is very difficult to learn, but it's critical. So I think, especially for the students, there are two things. You have to be alert for other technology, other fields, but also you have still to rely a lot on traditional, and that's why also Kurt is always insisting on having a microscope to be able to see the cells, because that's, and still I think the critical unit is a cell or the virus, whatever.
If you don't consider a cell.
No, I consider a cell. No, I consider, you have to think in terms of put yourself in the place of a virus. How are you going to interact with your host? I think that's, you have to think in these terms, not some kind of mush, that you put on a filter, and you saw the problem with filtering, It's,
So thank you, Daniel. So I think we are in time now. I think it's I don't know if anyone wants to make a comment. Sure.
No, I will.
Okay. To go back to the mechanisms of Darwinian evolution, I don't see why gradual variation, which takes place, of course, should be incompatible with the punctuated equilibrium. They could both be taking place. Because in the short term, there will be no transformation of one organism in another organism by gradual variation, and in the long term, it will happen. And at the same time, there could be events of genetic changes that will have a pleiotrophic effect, that that will accelerate the event, the evolution. And perfectly, I think they could both mechanisms could be operating at the same time. It's cool.
Sure.
While I was picking at, in the statement, a Darwinian approach versus the punctuated equilibrium approach, my question was, what is the mechanism of that process that brings together genes? And there one is left with the other of the Darwinian simplifications that made us think we believe something and we don't. And this is the concept of what is the mechanism of evolution. Darwin said natural selection. I say that natural selection, that's what happens after the evolution. Natural selection will fix organisms, but natural selection didn't cause whatever that collection of genes is that gives rise to the organism. So when you say natural selection is a mechanism, you're really sweeping the interesting question under the table. See what I mean by that? Natural selection doesn't say anything about mechanism of evolution. It says something about fixation of evolution, but not the mechanism of evolution. So just to clarify that.
It depends on what do you call mechanism, but natural selection is operating also. It will select.
Yeah, there's no question. It's part of the process. There's no question. But still, natural selection is what happens after the step of evolution that puts something onto the table which is new, which is then selected for or not selected for. But so the selection is certainly part of the overall evolutionary process, but not the mechanism whereby a new organism is constructed, whatever that means. Is that fair enough?
I think you should continue this interesting conversation during the barbecue this evening with a cup of wine. Maybe it could be a good topic to talk about today evening. Just to summarize, we talk about the legacy of Carl Booth today. Was a really important symposium that we have today. Was an honor to have all the speakers today and that we could have this conversation about the things that we think that are important, the things that science should face today. So thank you all to come today, and you are all welcome to the barbecue. Invitato.
Just an announcement. The bus will be expecting for us like what's the time? Like now.